Hi, and welcome you all to the morning session. Today, the topic for deliberation is molecular markers in aquaculture. Uh, uh, Ma'am, if you could give the presentation.
Shall I proceed, sir?
Give me 1 second. Today, we have the honor of having Dr. Jacqueline Pereira with us. Her area of specialization is fisheries biotechnology. She has around 25 years of experience in fisheries field. She has a patent, and I think she has 33 gen bag submissions. Ma'am, I hand it over to you. You can start with your lecture.
Thank you, sir.
Can you go to PowerPoint again
Yes, sir. Yes, sir.
and start the slideshow?
Okay,
Thank you.
sir.
You're,
Okay.
you're audible, visible, and your presentation is also visible.
Okay, sir. Thank you. Thank you, sir. Good morning, everyone, once again. Today's topic for discussion is molecular markers in aquaculture. What is actually know about molecules? Actually, now previously we will go for any diagnosis like that. If it is any molecular microbiology aspects or any immunology aspects, what we'll do, we will go for the traditional method. The traditional methods, it will take much time. For example, if you want to complete microbes for the entire diagnosis, it will take at least 2 weeks period for the entire study of the entire analysis. Like that, if you are going for immunotechniques also, it will take for the anti-serum development, raising of anti-serum and production of specific antibody itself required for 14 weeks period. So nowadays we have the molecular techniques. It is within 2.5 hours, within 3 hours, that is almost the complete confirmation we will get at the maximum of 4 hours period. So that is why the nowadays molecular study is very important, and that is let us see one by one. As you know, the molecules here we are going to discuss, that is DNA, RNA, then protein also. So DNA, RNA, that's these are the macromolecules, and this DNA level we can able to test. This is these macromolecules are DNA that is present inside the nucleus, that's present inside the cell. Each cell have our own DNA. That's all cells have the same DNA throughout the body, and this DNA is inside the nucleus, and inside the nucleus, that's a chromosomal DNA, and that is showed in the slide. And this why we are
Is it me or the frame has frozen from the speaker? Hello? I think our resource person got disconnected. She'll be joining soon. The resource person got disconnected. She's trying to connect. Thank you. You
Now,
can continue.
now it's okay, sir. Sorry, sir.
Yes, yes,
There is
yes, yes.
some connectivity issue. Sorry. Extremely
No
sorry.
problem. You're
Sir,
audible.
okay, okay. I'll continue. Actually, this molecular test, why it is required, that is need to, because if you purchase coming to our aquaculture field, before starting, actually we are doing that shrimp farming. Shrimp farming, it's the shrimp farmers, entrepreneurs, they will purchase a seed first. That's the first step. So After pre-stock management, they will start as a seed stocking. So when they purchase the seed, immediately after the procurement of the seed, they will, if they go for diagnosis, that molecular diagnosis will help to determine if any disease, even it is in the initial phase. Even it in the initial phase, it is possible to test within 4 hours, at the maximum of 4 hours, but within minimum 2.5 hours, like that also it is possible to give the result. So in that way they can minimize our time, energy, money. Otherwise that leads to great loss to the aquaculture industry. So in that way, molecular study, molecular diagnosis is going to help, molecular markers are going to help in such a way to prevent the loss for the entrepreneurs. So in this way, it will help to prevent the wastage of time, energy, and money. So let us see what are the areas of molecular markers based diagnosis. So here we can able to test the specific pathogen, specific species identification, specific organism. We can identify species specific. And not only this is applicable for the species identification, means not only for the microbes or virus or a bacteria or protozoan or fungus, it is applicable to the species identification. If it is the particular species or Savannah itself or something else like that, or the particular species of the fish also, not only applicable, restricted to shellfish, or if it will cover all pathogen, bacteria, and even the zooplankton, phytoplankton, all everything we can test. All biological organism, any organism can be tested. We can able to identify the genetic variation also. So area wise, the population wise, if there is structural changes will be there. So that can also be genetic modification now within the population and between the populations, that can be assessed. And the demographic, the bottlenecks of the particular natural population can be ascertained. And it can be applicable for the rehabilitation programs. It can be applicable for the disease diagnosis, food fraudulence. Some days, sometimes people actually, if it is in the frozen stage, if it is a live one, the entire organism will be there. If it is the processed stage, readily cooked stage, readily eat stage, that field also it is applicable. After making ready to eat or ready to cook, it is not possible to test, or it is not possible to identify this is actually of this particular fish or not. Or so if it is if they are doing food fraudulence, means only they are mixing 10%. So the mixing of 10%, it's not possible to identify by taste at all. So it is possible if with this our molecular test. So it will give best as well as appropriate result. So it is even in the disease condition, once there is any disease outbreak, then entry of new pathogen by some visitors entry, but any containers entry, anything that can also be, once we started the culture, it's going on culture. In the middle, we during the culture period, there is outbreak of some new diseases that can also be tested, and immediately, and we can give correct therapy. Therapeutic measures can also be taken in appropriate time is possible with molecular testing. So this slide shows the DNA structure. You know that actually DNA is a negatively charged one, so it will move towards the positive electrode during the. That's why during electrophoresis it is moving. This is the article for the molecular work. How it's simply loaded molecular identification methods. It is some of the articles. Then let us see the next. The technique is that's DNA fingerprinting. This is one of the molecular marker. DNA fingerprinting. We know about the fingerprints. Fingerprints usually in the older days, people they may not even not only the fingerprints older days, nowadays also our other card and else our fingerprints are available. For the each person to person, it will varies. So like that, our fish or shellfish also have this fingerprint. How it is possible? Finger, they know it may not have a finger like us, but DNA fingerprint. But the DNA is possible to give the accurate result. So it is helpful to the forensic test. Actually, if you fingerprint in general, it is applied for the forensic test. It is applied to the paternity test. Some of the male parent DNA, not in a real parent, maybe the child parent, the person we don't want to say, and the father. So such conditions he will deny. So that can also be detected by our DNA fingerprint molecular marker. Then already, as I told already, food fraudulence. Food fraudulence, if they mix for 5% or 10% of the particular species, so that can be tested. Actually, in our aquaculture field, blue crab, you know that blue crab. If they mix blue crab instead of blue crab, a certain percentage of blue crab they have reduced, and for that, if they are adding other crab meat like mud crab or spotted crab, that's sanguinolendus. So that leads what will happen? Simply by eating, we cannot identify. So this molecular test able to give whatever the percentage they have added. So molecular level is giving accurate and rapidly we can identify the fraudulence. And this again, fingerprints also helpful for the genetic variation in the among the stocks. We can that will give the polymorphism, so that we can it is possible to identify. So next we will discuss the another marker, that is RFLP. So before going for the RFLP or cloning methods and all, we need the restriction enzymes. Actually, enzymes, you know, that something, it will speed up the reaction. Here it is not like this, is something different. This restriction enzymes are nothing but they are the biological scissors. These biological scissors are nothing but it will cut at the specific site of the particular genome, specific site of the particular DNA. Actually, we know that we all, God has blessed with our own immunity. We have immune response. So innate immunity, specific immunity there. So our cell-mediated immunity, we have our own defense mechanism. So defense mechanism, why it is given? To protect us. So like that bacteria itself have its own defense mechanism to protect from the infecting virus. So that is phage, bacteriophage. So to prevent, to restrict the entry of bacteriophage, bacteria itself have the restriction enzymes. To cut the entry, to limit or to restrict the entry of the foreign virus. So that is why it is present inside the bacteria. So such external entry, that's entry of the phage will be restricted or limited by the presence of this enzyme. This enzyme, the scientist Werner Arber, he first found it is present in the bacteria. Then Hogan and Boyer, another group of scientists, they have thought, why not we can use this enzyme for our cloning experiment, that genetic engineering experiment. So these nothing but restriction enzymes are nothing but nowadays using as a biological scissors. It will cut at the specific site. So these enzymes have the enzymes always end with ASE. ASE means it will digest where it is. Such a way, restriction enzyme, it will cut, it will digest the nucleic acids. That is DNA, RNA, so it's nucleic acids. So if it cut at the specific site, so such a based on the location of the digestion, the nucleus can be classified into endonucleus and exonucleus. Endonucleus means that is if the nucleic acids in the internal region. Exonucleus, it will be in the end terminal regions. So it is a exonucleus activity. So there are different types of endonucleus, type 1, type 2, type 3. It has both restriction, type 1 is having both restriction as well as modification activity. It can able to cut the 1,000 nucleotides or more from the recognition site. For that, it requires ATP for energy. And the type 2, it has again the site of restriction activity. It also consisted with the adjacent to the next to the restriction site. So this type 2 requires magnesium ion as a cofactor. It's a cofactor. It requires magnesium ions. Then type 3, it is the restriction, again modification activity. This also similar to the type 2, it close to the recreation site. Here also it requires ATP for its function. Let us see the nomenclature of the restriction enzymes. Actually, it is the first letter, for example, ECO or 1. The first letter E derived from the genus name, that is Escherichia, from the Escherichia coli, from the Escherichia, E letter, capital E has taken. Then coli, CO, that is species, from the species CO has taken. Echo. Then R1. R1, I can give with example. R1, that is strain name, RY13. From that, R has derived. Then 1, Roman letter 1. So it was the first enzyme, endonuclease, isolated from the Escherichia coli. So Roman letter 1. Like that, it is ECO R1. Then BAM H1, HIND3. Like that, nomenclature was given for the restriction enzyme. Then let us see where exactly. Already I told restriction enzymes that will cut at the specific site. Specific site means, for example, BAM HA. So it will produce it will cut or digest it between GG. Like that, ECO R1 between GA. Then ECO R2, that is between, that's before CCT GG. Like that, HIND3, like that, all the enzymes. Then, if the restriction enzyme digest, we will get the smaller, the bigger DNA nucleotides will be, the DNA molecule will be convert after digestion. We will get smaller DNA fragments. So smaller DNA fragments, if that digestion by the restriction enzyme, we get two types of the product. One is with blunt end. Blunt end, you know that when the pencil is not sharp, you'll say it is blunt. So if it is sharp, here nucleic, the same way we are applying, it's blunt end. You can see the double strand will be there. Then sticky ends. Sticky ends, you can see here in the picture, in the bottom strands, it is TTAA, it's one sticky end, and another sticky end, AATTC. This is nothing but single strands getting by the restriction enzymes. And this single strand, why it is sticky? It is able to, single strands are not strong. It cannot withstand it. Otherwise, it have more affinity towards binding. So sticky nature, because the sticky nature, single strand is not stable. So it the sticky nature. That's why it is sticky ends. Then coming to the, let us see the other molecular markers, that is gel electrophoresis. Gel electrophoresis is nothing but, here the molecules get separated. Actually, during electrophoresis, the biomolecules, DNA or RNA, get separated under electric current. That is based on this separation is depends on the voltage or ampere of the current intensity, and the matrix which one we are using. Matrix, it can be agarose matrix or polyacrylamide matrix or nitrocellulose membrane, nylon membrane. These are the different kinds of matrix. Then buffers, which buffer we are using? It may be TBE buffer, it may be TAE buffer, tris acetic acid EDTA buffer, or tris boric acid EDTA buffer, like that. So for the electrophoresis markers, during that technique, we can load the sample like that. We have to first cast this is the submersible electrophoresis apparatus. In this apparatus, first we have to cast the gel, agarose gel. We casted here. Then while casting, we have to place the combs for electrophoresis. Then once after the agarose gets solidification, we can gently remove the combs and we can place the gel, casted gel into the tray. Tray is having the buffer. That is, it may be either TAE buffer or TBE buffer. So once electrophoresis is over, once placed, we have to give it for, it is placed in the apparatus, that anode cathode connected, and start the electrophoresis process. Once started, once it is over, once it started, that is based on the concentration of the agarose or polyacrylamide gel. So molecules, there will be intra-molecule space forming with. So if the density is high, concentration is high, molecules, the intramolecular space will be small, very small. If it is low, the space will be our intramolecular movement. Our DNA molecule have to move. So for the whole size will be high. So if it is, it can be 0.8 to 2%. of agarose. If it is 1% or 0.8, it will within half an hour, it will complete. If it is 2%, it will take 2 hours time for the completion of electrophoresis. Once the gel electrophoresis is over, so we can take the gel, remove the gel from the apparatus, and we can view under UV transilluminator or gel documentation system. So under UV light, we can able to see the DNA bands like that only. Here left side DNA marker is given. That is we can call it as DNA ladder or DNA marker. Then during the gel documentation, we will get pictures like that. Now again, this electrophoresis actually that depends on the different factors. One is molecular factor and another one is environmental factor. Environmental factor, we know that it's surrounding us. It's the environment. Molecular factor is belongs to the molecule which one we want to test. So that's based on the molecular. It is based on the charge, based on the size, based on the shape. These are the molecular determinants. It will determine the mobility, movement of the molecule. Then coming to the environment, it is the electric field, electric strength of the electric field, and the matrix already I have discussed. The pore size, if it is the concentration is 8.8, not 8, 0.8, it very less time. 1%, it will take around 45 minutes. If it is 2%, 1.5 to 2 hours will take. Then coming to the running buffer, which buffer we are using, what is the concentration we are giving. Usually will give 1% of concentration. Even for the running buffer, 0.5% also economically can be used. This is the electrophoresis apparatus. This is the another one. This is the vertical electrophoresis apparatus. Here we will call it as page polyacrylamide gel electrophoresis, or even denatured protein that is can be used. That is for the using SDS. SDS is nothing but sodium dodecyl sulfate. The other name of this SDS is sodium lauryl sulfate. We can call it as the two names. What is why we are using this? This is nothing but a detergent. This will denature the protein. Detergent will denature. We know that we are using detergents for washing our clothes and all, to denature the germs, to kill the germs and all. During the corona period, wash hands repeatedly with the soap solution or with the sanitizer. So here soap you are using for, that's a detergent that will denature the pathogen. So that's the same. In this way we are using here also detergent sodium dodecyl sulfate for the denatured ring of the protein. So the once denatured, it will get small fragments. So the small fragments, polyacrylamide gel, if the molecule is small, it will give better resolution, better results. Then coming to the types of matrix, the paper. It can be a paper, that's cellulose nylon membrane or nitrocellulose membrane can be used as a matrix. Then agarose. Agarose is nothing but so another matrix. Already I have mentioned. It is purified form from the agarose from the seaweeds. It is possible to extract. It's agarose is nothing but it's a polyacrylamide. And it is very, it will have based on the concentration, the pore size will vary, and it is frequently used for the DNA molecules. Then coming to the acrylamide gel, it is orderless crystalline solids. It's a chemical compound. It is that soluble in water, ethanol, ether, chloroform. It is used to synthesize polyacrylamide for many purpose. And again, another like this acrylamide, starch can also be used. Cellulose as a base also can be used. This is this picture shows agarose gel, electrophoresis gel. This here, you can see the different DNA patterns. That is the movement. Different molecules get moved, get separated in the gel. The larger molecules actually move slowly. If it is the molecule is small, movement will be faster. Lighter molecules move faster. Smaller molecules slowly. The two people running. If the fat people means for them, it's difficult to move fast. If the slim body, they can move fast like that here also. Once it's separated because of the electrophoresis, we can visualize under either UV transilluminator. If you are using the fluorescent dyes like ethidium bromide. Ethidium bromide is nothing but it is it is carcinogenic. Cautiously we have to handle, and it is a fluorescent dye. This will intercalate the DNA and it will get eliminated under UV light. So this is the polymerization of acrylamide shown in this picture. This is a very stable gel. It can be used for a variety of other purpose. Here the large varieties of pore size are there. Small varieties also, small molecules also can be detected with the polyacrylamide gel. Then coming to the techniques, that's blotting. Blotting is nothing but here the transfer of DNA from the gel to the solid. The solid like nitrocellulose membrane, nylon membrane. So we are transferring from the gel, DNA molecules from the gel into the nylon membrane or nitrocellulose membrane. So this method here, DNA, by this method we can transfer DNA, RNA, protein. We can immobilize into the nylon membrane or nitrocellulose membrane. If we are using, our sample is DNA, so that is that blot technique, we can call it as southern blotting. Why we are calling it as southern blotting? The scientist, he has given his own name to honor him. To honor his the scientist name, his name is Southern. So he has given his name for the particular marker, that is southern blotting. If once he has given it for DNA, it is southern, then later for RNA the term northern has given, then for protein the term western has given. That's why it is northern blot, western blot. Coming to the southern blotting, actually here the reactions blotting, actually it is why we are doing blotting techniques for the hybridization experiments. Hybridization is nothing but we are using different probes. Hybridization is we are different thing making together. Why we are using probes? Why we want to make together different? We want to detect our target organism. So target organism detected by the nucleic acid probes. What is probes? Probe is nothing but it will seek. Probe means seek. It will seek where exactly complementary sequence is there. The probe is single-stranded nucleic acid, and it will seek. It is short fragments, and it will seek where exactly complementary sequence is available. For example, if it is the template DNA have adenine A, the probe thymine. So thymine with the probe TAGC, like that. some short fragment of nucleic acid, it will search where exactly, or it will seek where exactly complementary sequence present in the target molecule. So that will be detected by the nucleic acid probes. So for that, our target DNA, our sample DNA should be extracted first, then for electrophoresis. After electrophoresis, it should be transferred into nylon membrane or nitrocellulose membrane, that is blotting technique, in which we are using the probes. So that can be, for the once we have used the probes can be labeled. It should be labeled for the detection purpose. It should be labeled with either fluorescent dyes. If we have the fluorescent microscope, fluorescent dyes like rhodomin, fluorescent, we can able to identify. If it is fluorescent, green light illumination will be there. If it is a rhodomin, it is a for that we need autoradiography instrument. So let us see some of the applications of DNA blots. Many restriction enzymes that cut, that's with the RFLP, with the RFLP technique, RFLP we are discussing later. So it is again with we can hybridize, we can use probe with the blotting techniques, and we can identify closely related individuals, and we can confirm the gene transfer or a gene restriction. We can detect the foreign DNA. If actually most of the genome, with even the microbes also, DNA as a genetic material. But some virus, like HIV, it is RNA virus. Its genome itself RNA. So in such conditions, we can go for RNA blotting. Not only that kind of virus or that pathogen, even for any genes expression, for the human side or our fish side, any gene expression, it will be expression. DNA will be transcribed into mRNA. So gene expression will be detected by the RNA. So RNA, once separated or extracted or isolated, that can be denatured into the gel. Then from that, it can be transferred into the nylon membrane or nitrous membrane, then followed by hybridization with the probes. So it will give the detection on the film with the fluorometer, either fluorometer or with the radioactive probes with the help of autoradiography. Then coming to the application of RNA blots, that is already I mentioned, for the detection of gene expression levels of the particular transcript. And even it is some specific genes of some genome itself RNA, so that can also be detected. Then coming to the Western blot. Western blot is meant for protein. So protein, it's a blotting. It is give highly specific qualitative test, and this can be detected with the SDS page. Already I told, page polyacrylamide gel. Then it is using SDS, sodium dodecysulfate. We will call it as SDS page. So here only the difference is, instead of a DNA or RNA, we are using protein. So how to load the sample? That's in the polyacrylamide gel already we have seen. Then coming to the hybridization part, actually the probe we are using, that it can be DNA or RNA. So such hybridization can be DNA-DNA hybridization, or DNA-RNA hybridization, or RNA-RNA hybridization. Then even protein also, protein with the protein. So any protein or that have the specificity, so that can also be detected. So it just depends on the extent of complementation, how it is complementary. If adenine thymine, it's a complementary one, like that. CG, cytosine, complementary to the guanine, like that. It depends. It depends on the concentration. Then again, it depends on the temperature, salt concentration, and solvents. So temperature also important, and salt concentration also important, and the solvents in which we are diluting. So these all the factors determine. And again, any factors like charge of the thing, electricity, and probe, which are radioactive labeled one, fluorescent labeled one, enzymatic labeled. So these, and if it is used with the dyes like ethidium bromide, like that, the probes again depends on the probe sequence. Also, it will vary. Then again, this hybridization, that is the in situ hybridization. In situ hybridization that is performed by denaturing of DNA of the cell squash, that is on the microscopic slide. So once we have prepared the microscopic slide, the squash, that could also be within the cell itself. That is why it is in situ hybridization. We can use the probe within the cell. It need not go for the DNA extraction or RNA extraction. Within the cell itself, it will hybridize and give. So here we can use DNA probes for the detection of the chromosome, on particular chromosome, and then that will give the particular genes. Then like that we can give, even in the tissue can also, chromosome in situ hybridization as well as tissue in situ hybridization also there. So here the RNA probes can be used within the cells as well as tissue for identification of gene expression. This is possible with the hybridization. Again, oligonucleotide probes. Oligonucleotide is nothing but ATGC. So this combination, this single-stranded one that can be used to hybridize the amino acids, to predict DNA sequence, and to find out the combination, how far it is have the hybridization. So it is this gives specificity of the single nucleotide. The changes can also be detected. Is any point mutation occur, that can also be detected in the probes. So the probes already we have mentioned. It is radioactively fluorescent dyes with autoradiography. If you are using a radioactive carbon, autoradiography. If you are using fluorometers or fluoroscanner or fluorescent microscope, fluorescent dyes. If you are enzymatically method with the HRP, like that, we can use different dyes that will give the ELISA radar, like that, it will give the color emission, light emission can be detected. Then next molecular marker is PCR. PCR, actually many of them know about PCR. PCR is nothing but polymerase chain reaction. So this is a novel technology. Actually, because of the corona, even the laymen know what is PCR, because this was we could able to hear the term, even the PCR in the news. We always precautionary measures are given for our government has given precautionary measures, alert has given about corona. So during that period, people know about the PCR. What is PCR? PCR-based test was carried out to diagnose the coronavirus. So it is this technology was discovered by the scientist Carey Mullis during the year 1983. After 10 years period, he got a Nobel Prize for his discovery for the in chemistry. Actually, he was a biochemist, so he got a Nobel Prize for his discovery. And this polymerase chain reaction can be carried out using a thermal cycler, that we can call it as DNA engine or DNA machine, or DNA PCR thermal cycler also we can call it as. So this in this slide shows PCR thermal cycler in the picture. Actually, this PCR thermal cycler, the picture shows all the steps in the picture here. So first step you can see here, the DNA extraction. Once extracted DNA, immediately we can use. Otherwise, if we want to store, means we can store either -20 or even deep freezers -85, -65, like that also. So PCR records once it is extracted. So it records the various compounds like DT, Taq DNA polymerase, DNAPs, and it has various steps like denaturation. Actually, there are 3 major steps involved in the polymerase chain reaction. That is denaturation. Denaturation is nothing but DNA is double-stranded one. It gets denatured into 2 single strand. Actually, these things are not we are will see in the pictures in the coming slides. Then the next step is annealing. Here you can see the primer when it is bind with the template DNA. That is annealing step. Third step, extension. That Taq DNA polymerase extend the DNA synthesis process. So at the end of one cycle, from one molecular nucleotide, that will be produce 2 double strands. So like that, if we are using 30 cycles, 2 power 30. 2 power 30 means 2 power 2, 4, 2 power 3, 8, 16, like that 32 millions of copies we will get at the end of the 30 cycle or 35 cycle. That will vary. It's not a fixed one. Based on the particular genome, based on the primer, based on the literature, we can design the PCR program. So here this picture in the polymerase chain, this picture shows polymerase chain reaction. There are different steps: denature, annealing, and extension. This picture here itself, you can see the documented DNA after electrophoresis and after the UV gel documentation system, which is possible to document. So here, this step, it will show the PPT slide shows steps involved. The first step is DNA nucleic acids separation. So once separated, the target DNA gets separated. When it's separated at high temperature, when the temperature is 98, 94, during that temperature, DNA gets denatured and it gets separated into 2 single strands. For that, again, we are using the magnesium ion as a cofactor. So you can see here at a high temperature, that 94 degree and 95, 98 degree centigrade, it is a double strand of the DNA is possible to denature into 2 single strands. Once it formed single strand, then only the priming means initiating. It will initiate the the steps of synthesis, the complementary DNA synthesis. So for that, it requires DNDPs, tag DNA polymerase. So the step primers are nothing but single-stranded nucleic acids. So single-strand nucleic acid, it will bind like a probe. Already we have discussed in the hybridization. Here, this is also short fragment of DNA or short fragment of nucleic acids. It will bind where exactly complementary sequence is there. So it will bind always in the 3' end. So 3' end of this DNA synthesis will happen. So, here you can see this polymerization step, that is, or the extension. This step, otherwise call it as extension or elongation. This extension or elongation will be carried out by the enzyme tag DNA polymerase. Why tag? Actually, DNA polymerization is happening. For that, the enzyme required is DNA polymerase enzyme. DNA polymerase is nothing but it's an enzyme involved in the polymerization of DNA. But all enzymes are proteins. All proteins get denatured at high temperature because we are using in the thermal cycler, in the PCR reaction, high temperature for denaturing of protein. The denature of protein requires high temperature. So this high temperature leads to the denaturation of our uh, DNA polymerase enzyme also, not tag DNA polymerase, DNA polymerase enzyme, because it is a protein. Once it is denatured, it cannot work well. It cannot work. So it will not be in the light, lose its ability. So to solve this problem, the scientist has extracted the DNA polymerase from the bacterium Thermus aquaticus. It is extracted from the hot spring. Hot spring, it is extracted. So this Thermus aquaticus can withstand even at high temperature. So that now the tag DNA polymerase enzyme was extracted from the Thermus aquaticus. So it can withstand even at very high temperature like 98, 94, 95. So that is we are applying in the PCR reaction. So that is why we could able to do successfully polymerase chain reaction with the help of tag DNA polymerase enzyme. So we can able to multiply millions of copies. How it is able to multiply? That is approximately 150 nucleotides per second. It is undergoing the process of polymerization. Then let us see how PCR works. So many of the points we have already discussed in the PCR works. It is oligonucleotide primers we are using. So with the primers only, priming the reaction, initiating the DNA synthesis. And
Hello, sir. Hello. Sir, shall we leave the hall, sir? Sir, shall we leave the meeting hall, sir?